Hi, everybody. I am Michelle Tennant Nicholson. I am the current president of the Henderson County Beekeepers Association in beautiful Western North Carolina. Basically, the Smoky Mountains is what you've probably heard. So I went to conference with the North Carolina State Beekeepers Association a couple weeks ago, and on stage were all these beautiful women. And I was like, yay, women in beekeeping, hell yeah. And they were on stage with Dr. Tarpey and they've been doing a lot of stuff. So I put the word out like, hey, let's get some of these presentations recorded for YouTube so that we can actually share with the world what's happening under Dr. Tarpey's tutelage, and then some of this beautiful research that's happening. And Tejal is somebody who accepted my invitation. And Tejal, I'm just going to let you introduce yourself. So, and then what you're going to talk to us about today, which is the presentation that was given at conference. Tejal?
So thank you so much, Michelle. I am very honored to be here, very
Thank
honored to
you.
present this research as well. So I'm a student from Virginia Commonwealth University, and I am in NC for this summer researching about bees and microbes under Dr. Tarpey's tutelage. And what I did was I figured out, I'm going to read this really long title, which feels really long, but I started it's really, really easy, like once we start going through this presentation. So deciphering abundance, diversity, and symbiotic roles of prophages within bee gut microbiomes.
That's what she said.
So all that means is I'm checking out those little bacteria that are inside the bee guts, and what is inside those bacteria is the prophages, which is like tiny little viruses, which can benefit or harm our bee health or bee fitness. And I'm just seeing how that affects colonies across North Carolina, like bee colonies across North Carolina. And I did this with Yusuf, who happens to be my tutor. He's a postdoc here, and Dr. Tarpey, of course. We all love him.
Yep.
All
Well,
right.
hold on. They may not, right? If you don't know that, you can go to ncbeekeepers.org and you can click on endowment. So for beekeepers in North Carolina, we are all fundraising to make sure that the endowment moves forward. And then you can read more about Dr. Tarpey there. Go ahead, Tejal.
All right, so if you're ready to get started, I can
Yes.
head on to the next slide.
I actually walked in when you were halfway through when I was in the bathroom. So I'm so excited to actually see yours from the beginning now, right?
Let me slideshow this.
Yeah, and then when you're in the slideshow, I think you can just hit your forward arrow. Okay.
Yeah.
Tell me if it doesn't, if you need help with that. Go ahead.
All right, sounds good. So here particularly, I'm looking at two different bacteria that are inside bee guts. We consider this the core gut microbiome, so it's highly specific, and it really shapes how bees behave and how their fitness changes over time. So I'm looking at Gilliamella and Snogrosella. If you've heard, we are what we eat, that's basically what this is, that we are what is in our gut. So yeah, I'm looking at these two bacteria. I'm looking at the prophages that are found inside. So we can think about prophages as basically probiotics or prebiotics that are in there that help us with digestion and stuff. So yeah, that is what basically we're looking at in this research. All right, so how do we examine this? So first we need to find out what is inside the gut, right? So I get all the genomes of the bacteria that are inside these guts of honeybees and bumblebees. And these are usually just raw input from the NCBI where we find these genes and genomes. And then what I have to do is, since these are bacteria, I want the specific ones that are viruses, which are prophages, which usually benefit the honeybees and their health. So what I do is I use this three softwares, which is VirSorter, CheckMe, and Vibrant, to annotate those genomes and cut out the bacteria part and really just remaining probiotic, like the prophage part remains, which because so that we can find out like how it's benefiting the bee. in
And
general.
Tejal, is this information coming from beekeepers themselves in each of these sources?
So these are scientists. These are scientists who have sequenced the genomes of these bacteria, which were hosted by the bees. And
Wow.
these
Okay.
scientists have been like all around the world. So
Thank
um,
you, bee scientists.
yeah, we love them. We couldn't do this without them. And because of this, what we get is prophage abundance, diversity, and function. So abundance is how many prophages we have in a bee. Diversity is how many types of prophages, how many different types of prophages we have. The more diverse the environment, the better it is for the bees. And then the function. So what function does each of these prophages serve in improving bee health?
Okay, great. Okay.
Mhm. So this is what we have found. So we have found that we did a correlation between honeybees and bumblebees, um, and saw how their prophages, like the total prophages in them, vary. And then we also did a box plot across species. So the Gallimiella apicola, apis, they're all the bacteria that are found inside bee guts. And then we found out like the total prophage concentration and how it varies across each bacteria. Um, and this is between honeybees, and the bottom one that we see is between bumblebees. And we found out that there's significant difference between Gallimiella sp and Snogrosella albi, which are completely two different species, correct? And we can see clearly how much one is like how much more one hosts compared to another. And then we also found out the correlation between genome size and the total hallmark. So that means total hallmark means a viral protein concentration. So that means we see a slight positive correlation between the viral concentration and genome size. So as the genome size of the host, which is our bacterial host, which are inside the bee gut, increases, our viral protein concentration also increases. And we can see the same bit amongst the total phage, so the total prophage. So as the genome size of the host, bacterial host increases, the total phage concentration also increases. And it is important to know this because we want to find out how it is affecting the gut health, because we do see that there's a slight positive correlation here, right? And now our job is to find out that, okay, so if we go further with this, is this something that we could develop into a phage therapy? So something that could pinpoint a precise medication for a for something that's affecting bees. Like, suppose this prophage can cure. I don't know if you've heard about phage therapy, but it is like a precise medicative treatment for that is being researched amongst not really well amongst the veterinarian side of the field. And now it's time to find out, like, maybe we could do this with bees as well and see if it's applicable.
So for so one of the things that I know that people are going to be saying is can you what about the everyday language here? So the everyday language here,
Mhm.
Tejal, because it's very academic. I love it. I have a master's degree, so I'm like I'm
Right.
into it. I'm into it. But for the everyday beekeeper, what this means is strong, as like you have a strong hive. The best way to deal with pests in your own hives is to have strong immunity,
Exactly.
period, end, right? So full stop, that's what we're looking for here, and that's what Tejal is working on. Great. So we're looking at like what is affecting the bee gut health so that we can, as beekeepers, really impact that total health and immunity of that hive. Okay, what's next? What else did you find?
Exactly. So, yeah, so that is what we have so far, because let me tell you, we were given 2 months to do this research. And research is a very organic process, but now it is our job to find out how, if it's going to positively affect or if it's going to negatively affect. But we have an idea that it does make an impact on the gut health. So, but thank you so much for putting my words in the everyday lingo. Yeah, like I definitely agree with exactly what you said, that that is exactly what we're doing here at the Tarpey Lab.
Well, and then Tejal, those beekeepers, from what you found, is there anything that we should, it's like, should I be feeding my bees probiotics, or do they just naturally get it in the field? Do you have an opinion on, you may not have an opinion on that because I know it's highly controversial, but
Mm-hmm.
what would you advise us as average day beekeepers? Anything?
As in, so considering like my research is in progress, so I definitely am not in a position to give you an opinion. Um, but I do feel like it is important to emphasize on emphasize on all over perspective rather than just like um, giving something that's not researched. Like that's definitely not the way to go. And definitely like try a holistic approach when we're looking at something that's affecting bees and beehives around the world.
So, just to kind of end powerfully, because this is your last slide, right? This is what you
Mhm.
found.
Yeah,
One of the things because we had a few other speakers on this at our club, and
yeah,
I've been doing my own research, and it's just like your own gut health, everybody.
right,
You want to have diversity in the food around you as much as possible. It's really like what makes uh, the bacteria, what's inside the gut, really happy.
right.
So one of the things that we can do, and it's not contested in science, is having a diversity of plant life around your beehives. So if it's not naturally occurring inside your environment around you can plant it, and you can plant different flowers, a diverse array of flowers through the year. And I
Mhm.
highly recommend that you look at that so that you're really focusing in on the gut health of the bee and then also the immunity. And Tejal, uh, we're not going to talk about some of her new studies because she has some really wonderful things to share in the future with us. And I just want everybody to uh, remember her name because I think that she's going to be making another contribution to us in the future. And thank you so much for spending time with us today.
Thank you so much for having me.
Bye bye.
Bye.